Okay, hey everybody. I'm going to read this article in case you'd rather listen to me read it than read it yourself. So here we go. I'll try to highlight along where I'm reading. So the article title is Bite Mark Analysis Junk Science on Death Row for a Crime He Did Not Commit. In the early 90s, Kennedy Brewer was convicted of killing a 3-year-old girl. Brewer was sent to death row. After 15 years in jail, modern DNA technology proved that Brewer was innocent. The actual murderer was a man named Justin Johnson. So, how did this happen? Very little evidence was left at the crime scene. Brewer's conviction hung on the testimony of an expert bite mark analyst named Dr. Michael West. Dr. West said he was certain that the marks on Jackson's body came from teeth, and that those teeth, indeed and without a doubt, belonged to Brewer. So here, pause and answer in this box, what evidence convinced the jury that Brewer was guilty? The problem with bite marks. It's hard to determine the source of marks on a body because what we're looking at, or quote, what we're looking at isn't actually a bite mark indentation. What we're looking at is the bruise that's left over, Dr. Freeman explained. Freeman went on to explain that bruises often take a different shape than the teeth that made them because blood pools and spreads out under the skin. Furthermore, some people bruise easily and others don't. He added that skin is elastic, and this elasticity varies depending on how much fat a person has in their body or how old they are. Skin also holds tension and releases in different ways as you move, which would affect the impression teeth might leave on a person. Pause here and answer. Freeman listed variables that would need to be kept constant to make bite mark experiments fair. Name 2 or 3 of them. This means that if the same person bit 2 different people, the impressions or bruises they would leave wouldn't necessarily match, and the way that these factors impact bite marks would never have been studied. And the way that these factors impact bite marks have never been studied among living people and likely never will be. Dr. Freeman explains that people bruise differently depending on diseases and skin tone. Some of those people are going to have cancer or diabetes or sickle cell anemia. Some of those people are going to be black and white and Greek. Pause and answer. Why is it probably impossible to run a fair, controlled bite mark experiment, and that would be needed to make bite mark analysis actually scientific? And that's the end of part 1. Here's part 2. Bloodstain pattern analysis, flawed but improving. Wrongfully imprisoned based on faulty science. David Cam lost 13 years of his life to 8 drops of blood. In 2000, David Cam, an Indiana state trooper, was charged with murder after finding his wife and 2 children shot to death in their home. During the 3 trials that followed, the prosecution brought in bloodstain experts who argued that the flecks of blood on the t-shirt Cam had worn the night of the murder were high-velocity impact spatter, proof, they said, that he was the shooter. On the other hand, other experts called by the defense testified that the blood was actually a transfer stain that had smeared onto Cam's clothing after he tried to help his children. Nevertheless, Cam was sentenced to 195 years in prison. He remained behind bars until his acquittal in 2013, after the conviction of another man whose DNA had been found at the crime scene several years before. Cam's case is only one of many that have come under fire for their heavy reliance on bloodstain pattern analysis, a subfield of forensic science criticized in a report released by the National Academy of Sciences in 2009 for being more subjective, opinion-based, than scientific, and filled with enormous uncertainties. But bloodstain pattern analysis might be in the process of changing, thanks in part, perhaps, to the work of a team of physicists that's combining the study of blood spatter with fluid dynamics. Their latest model, published today in the journal Physics of Fluids, can now accurately match a bullet of a certain size and shape to the size, number, and speed of blood droplets produced by a gunshot wound. Pause here and answer. What's the main idea of the last paragraph in the section above? The shaky foundation of blood spatter analysis. Some of the methods that blood spatter analysts have used in the past to figure out the trajectory of the bullet and point of origin of a stain may have been too simple, leaving out some of the complexities of how blood moves and changes after leaving the body, says Daniel Attinger, a mechanical engineer at Iowa State University. Part of the problem is that blood isn't an easy fluid to work with. Liquids like water flow at the same rate no matter how hard they're shaken or stirred. But the same isn't true of certain mixtures like ketchup or blood, which actually get runnier if more force is applied to them. As a result, blood drips slow but sprays fast. Blood is also incredibly changeable. It teems with living cells and active enzymes, and its properties shift under the influence of minute fluctuations in temperature or the presence of drugs. It's the most complex fluid he's ever studied, Attinger says. As soon as it leaves the human body, blood begins to cool and thicken while also warping under the combined influence of gravity and drag from the air it's moving through. That's all before blood meets the surface it ultimately lands on, the properties of which can seriously affect the impressions left behind. If blood's landing pad has some bounce, it will affect the way the fluid splashes. If it's rough or full of holes, it will change how the blood distributes itself. Pause here and answer. What are some variables that make bloodstain pattern analysis tricky and that have not always been taken into consideration by past analysts? Thicker than water and far more complex. This gap in knowledge is where fluid dynamics come in. Many of the complexities of blood in motion can be captured with equations. Attinger and his colleagues have spent years crunching the numbers, coming up with increasingly complex models that account for more and more of the blood spattering process. The team did their fair share of hands-on controlled experiments too, firing bullets at everything from sponges soaked in pig's blood to suspended sacks filled with the real stuff taken from human bodies. After collecting the spatter on clean, smooth surfaces like butcher paper, they'd see how the results of their computer simulations stacked up and adjust accordingly. The researchers' models can now demonstrate a mathematical relationship between the shape and speed of a bullet and the patterns of the resulting blood spatter. Pause and answer. What are Attinger and his team of scientists doing to make bloodstain pattern analysis more fair? Attinger acknowledges that these models and scenarios are still oversimplifications. Sponges and sacks of blood aren't human bodies, and the walls and floors of crime scenes aren't usually lined with paper. The different types of fabric used in clothing also present a complication that hasn't yet been accounted for in crime scene reconstruction. Still, controlled experiments like these, in which researchers can control and keep track of all the main variables at play, are necessary starting points, he says. Pause and answer. What variables do scientists say have not yet been investigated via control experiments? The researchers hope that their models will someday become sophisticated and realistic enough to be translated into a chart that spatter analysts can use in the field. An even loftier goal, the scientists say, would be a cell phone app or handheld device that could scan photos of stains and spatter, crunch the numbers, and spit out a likely rundown of what happened: the type of gun, the bullets used, the locations of people involved, each with a score of the algorithm's confidence in its results. Pause and answer. What is the long-term goal for these scientists? Much of the scientific gap in these disciplines is about communication rather than lack of effort of any kind or of willful ignorance. This knowledge won't do any good if it's not made available to blood spatter analysts who act as investigators and expert witnesses. The fact remains that most experts in the discipline haven't been trained to interpret research papers grounded in fluid dynamics. Tweaking training procedures for blood spatter analysts or encouraging academic researchers to join forensics labs might make a dent in these issues. But more sweeping reforms will be needed to actually put research like this in a position where it can have a real impact. But change will also require bringing other relevant voices like those of judges and attorneys into the bigger conversation. If judges and lawyers don't know about these advances, they may still admit out-of-date science in the courtrooms. That could land innocent people in jail. Pause and answer. What are some reforms this article suggests that may make the justice system more fair? All right. Hope you enjoyed it.